All right, so I'll talk about this HIF-2α inhibitor for renal cell, and certainly has successes and failures, which is quite interesting. As you have seen the news, April 21st, Merck announced that their phase 3 study, LITESPARK-012, actually failed. Now, this is a trial design here, the three-arm study. Basically, the arm C is the control arm, which is Keytruda plus Lenvima, which are the current standard of care for the first line treatment of renal cell carcinoma, which is a clear cell type. Now, renal cell, as you may know, has about 435,000 new cases per year globally. So it's actually not a small disease. And the treatment really has evolved quite a bit. But in this study, what happened was they tried to do two things, right? So arm B is actually their CTL4 molecule in combination with the Keytruda. It's a new formulation of the two molecules together, and in combination with the Lenvima, which is the anti-VEGF. And also arm A is which is quite interesting because it is a HIF-2α inhibitor, Belzutifan and Belzutifan. Now this molecule is has been approved already for renal cell cancer for a few years, and did quite a few studies. And this is one of the LITESPARK trials, and it turned out to be sort of a disappointment after some very, very good, successful trials early on. Now, I want to mention, just to remind everybody, the trial design. So number one, the control arm is the PD-1 plus anti-VEGF, and the other two arms, one is to add a CTLA4 antibody, and the arm A is to add a HIF-2α inhibitor, right? So it's sort of an SOC, so A plus B, A plus C, and A plus sort of compared to A, right? If we see A as standard of care. So it's a typical trial design in order to get approval of a new agent. Next slide. Okay, so this is a quick sort of overview of renal cell cancer involvement. Actually, you can see from the very beginning, really, renal cell is kind of interesting. It does not respond well to chemotherapy. So very early on, it is the high dose interleukin-2 as the main therapy for many years in the 1990s. And then the anti-VEGF emerged, the sorafenib, regorafenib, sunitinib, and then mTOR inhibitor came out. You have quite a few molecules. Then you have anti-VEGF, the bevacizumab, interferon, and other sort of anti-VEGF kind of molecules. Then you have a cabozantinib, then you have Lenvima. So at those time, I remember there was an article in JCO that says, well, the embarrassing richness of renal cell cancer treatment, right? Which is kind of interesting. The comment was that, wow, for renal cell, suddenly from nothing to so many new treatment at the time in the 2015 time frame. Then you have this immune therapy came out. In the 1990s, the IL-2 efficacy seemed to suggest this tumor is responding more to chemotherapy, to immune therapy. So Nivo got a approval in 2015 for renal cell. Then you have a Keytruda approval, and then you have CTL4, and finally you got this combined use of anti-CTL4 and PD-1 for renal cell, and also the approval of Pembro, so Keytruda plus lenvatinib, right? So this is really the standard of care for now. And then you got this brand new class of agent, the HIF-2α inhibitor. I think this is to me, it's very interesting. I remember when I was Amgen in the 20 sort of before the 2010, Amgen was actually working on the HIF-1α pathway, HIF pathway, and it did not pan out. At that time, there was a sort of a theory it might work, but I guess the molecule did not work out. So it's a pretty interesting evolving field. Next slide. So here's the quick mechanism, right? So it's your HIF-1 alpha, HIF-2α. Now HIF-2α is something that really is highly expressed in the renal cell, renal tissue, right? So that's one maybe reason why it could work for renal cell cancer. So under the hypoxia condition, you have increase in HIF-2α, which binds to the HIF-beta, and then it stimulates the transcription and the synthesis of things like anti-VEGF, GLUT1, EPO. So please remember this molecule. Right? So one is the VEGF, which is under angiogenesis pathway, and also EPO, which of course is important for red cell production. And this is very relevant for the efficacy and the safety of this molecule. So the way it works is that things like Belzutifan binds to HIF-2α and block the binding of this molecule to the transcription element on the DNA, which is sort of a mechanism of how this molecule works. Next slide. So basically, these are some studies that's going on with the Belzutifan in renal cell. I put a box on the phase 3 trials, the LITESPARK-011, LITESPARK-012, and the LITESPARK-022. So as you've seen in the early this year in the ASCO GI, the two studies, 011 and 022, are sort of successful. But this new one just came out, the news of LITESPARK-012 failed, right? So let's maybe quickly, the 011 is a second line study after IO. The 012 is a first line study, and it's in competition with the IO. And the third one is actually in adjuvant setting. So it's much earlier than the first line with this patient who have undergone the surgery for renal cell. And the adjuvant setting, they were given, and the standard of care is simply pembro, and this is a combination with this HIF-2α inhibitor. So you can see here the three big phase 3 trials, 2 succeeded, 1 failed. Next slide. So quickly, sort of for a recap of the results. So for 012, really, this is Belzutifan, a renal cell after failure of a prior IO therapy. So basically, it's a second or third line study. You can see the design is quite simple, and the control arm is simply the standard of care for that setting, which is cabozantinib, and the treatment arm is Belzutifan plus lametinib. So this simple trial design on the right side is a PFS was significant with a 0.70 hazard ratio, so 30% decrease in the risk of progression or death. On the lower left panel, it's overall survival. There's a trend, but it did not reach statistical significance. But the trend was there. You can see 0.85 hazard ratio. So that's slight decrease in the risk of death. But the number of events is kind of low, so it's not possible to reach statistical significance with this number of events. But anyways, this data is good enough to gain, I think, approval for this indication. And as you have seen, Merck has submitted the NDA for this molecule. The response rate is also higher on the lower right panel. You can see here the summary of the PFS, OS, and OR. OR is actually 52% compared to the control arm, which is lower. Now, so here, second, third line patients, the trial was successful. Next slide. It's kind of a delay in the, okay. All right, so this is the thing in the adjuvant setting, LITESPARK-022, right? This is also a very simple trial design. You can see it's a big study. So 921 patients compared to 920 patients. So very big phase 3 trial, but a simple design, which is the pembro compared to pembro plus Belzutifan, right? So very simple trial design. And you can see on the upper right panel, it's a 0.72 hazard ratio, and it's highly significant. So this trial is successful for the adjuvant setting, which is actually very good because it's a large patient population. And on the lower left panel, overall survival, the difference is sort of smaller. You can see it's 0.78 hazard ratio, and p-value is not significant, but for the adjuvant setting, this is actually quite understandable. That's normally this situation. So disease-free survival. Sorry, on the right upper panel, it's a disease-free survival, which is actually good enough to gain approval of Belzutifan for this indication. The one thing I want to point out on the lower right panel, you can see here anemia being a major adverse event for this molecule. It's much higher compared to the control group. So the top sort of a bar on the left side. And this is certainly on target dose-dependent effect because, as I mentioned earlier, the HIF-2α is a major sort of regulator of the production of EPO. So EPO decrease certainly will cause a decreased production of red cells. Another actually very frequent AE is hypoxia, which is also on target because the EPO causes decreasing red cell, and also it blunt the oxygen sensing. So at the end, the inhibition of HIF-2α will cause anemia and also the hypoxia. So these are on-target effect of the molecule. Next slide. So just quickly, so this is the clinical development of Belzutifan. So I think it's very interesting to see how the big pharma develop their molecule. I think it's sort of for all of us, we can really learn something, look at how they go about. First of all, the HIF-2α is certainly relevant in this VHL-related disease. So they went there first. So they got approval in 2021 as orphan drug designation for this disease and got approval. Now you can see the phase 1 trial started in 2016. So, and they got approval in 2021. That's about 5 years later, which is actually pretty good. And then they started multiple studies in very big indications for renal cell. They are also doing studies in other tumor types as well, but the renal cell certainly is the right indication to select for this molecule because of the expression of HIF-2α is mainly sort of in the kidney tissues, and also it's important in the regulation of the angiogenic pathway that is something relevant for renal cell as well. Next. Okay, so maybe some perspective, right? So first of all, Belzutifan certainly is practicing changing for renal cell. It is a brand new class of molecules. Now it's shown to have activity in second line and even later setting, and plus also for the adjuvant setting. So it's really going to be a big molecule, very important treatment for renal cell carcinoma. Unfortunately, as you have seen here, the first line study failed. Now, why that happened? I think I have to acknowledge, right? We don't know exactly, but some speculation for me. I think the standard of care first line is PD-1 plus anti-VEGF. Right? So you have pembro plus lenvatinib, lenvatinib as the standard of care, which is already pretty strong. So on top of that, to be able to provide additional benefit might be difficult to begin with. Number two, the HIF-2α pathway, right? One of the major driver for the benefit may come from the inhibition of the angiogenic pathway, because the VEGF is a downstream sort of effector protein of HIF-2α. So by blocking that, then you add additional anti-VEGF Lenvima, there's overlapping and redundancy of mechanism. So that might be one reason why this addition of HIF-2α to the standard of care, which is doublet of pembro plus lenvatinib, did not produce additional benefit, right? So that's one potential reason. And number two, even if you do have some potential synergy because HIF-2α has additional effect other than or in addition to the VEGF pathway, those benefits may not be sufficient to demonstrate a statistical superiority. Now, we don't have the details, but hopefully when we see it in the future, we can see if the two curves are overlapping or if there's any difference between the two. For CTLA-4 antibody, I think it's interesting to note it did not work here. Two reasons. One is that you already have a strong control arm, and adding CTLA-4 may not help or may not be sufficiently helpful. Number two, I'm not sure their dosing regimen, 25 milligram every 6 weeks for CTLA-4 antibody. It's unclear what would be the right dosing regimen. If you can see for ipilimumab from BMS, right, their dosing regimen in combo with the Nivo is quite different from this. And you have also from AstraZeneca, they were dosing something like more than 300 milligram, a single dose in combination with the Durva, and that was for liver cancer, right? So that's a very unique dosing regimen in that case. So it's for this molecule, it's dosing sort of 25 milligram every 6 weeks. Is that the right regimen? I'm not sure, but although they did mention in non-small cell lung cancer studies, they were able to produce efficacy in that regimen. Now, overall, I think there's also, in my view, a ceiling effect perhaps for renal cell cancer when you have a pembro plus lenvatinib. Whatever happened is already pretty good results. So the bar is very hard. The bar is very high. So it's difficult to overcome that bar in the first line setting. Now, this to me is another example of the challenges in drug development. You can win in some cases, but not in all. Next slide. It's very slow. Okay, even though there's quickly, you can see here in the Nature Review Drug Discovery, you can see the pipeline for molecules for renal cell. There are a lot of studies that are still ongoing, and the cells you can see on the right panel. So the forecast of the major increase in cells for renal cell would come from HIF-2α. You can see, right? So it's 27, it might be almost $1 billion. And by 2032, HIF-2α would add additional $2.1 billion sales to renal cell market. So that's pretty significant. And that prediction actually is before the results came out for these two successful trials. So I believe it's possible the forecast may be even an underestimate in my view. So that's what's for the future. Maybe 1 minute quickly, if you go to the next slide. I want to mention this combination of a pembro plus lenvatinib, so it's called a LEAP. So there's a LEAP program. There are so many studies here listed, right? You've seen this before. So there's a huge program. I think it's a very, very ambitious, very costly combination of the LEAP program. But what happened? Next slide. So all this big LEAP program, you can see on the right panel, on the right results, almost all failed. Okay, only trial that was successful was renal cell cancer with this LEAP regimen. So this is unbelievable sort of a cost. If you look at all the studies with the LEAP, it is very, very sort of disappointing, and I would say heartbreaking for the companies that sponsored so many big studies that all failed. Okay, I'll stop here.